Hello everyone, welcome back to the class. So yesterday we learnt about ribosome. Today we will be learning about endoplasmic reticulum. So let us begin the class. As I said, ribosome is a protein synthesis or protein factory of the cell. It produces different types of proteins. So endoplasmic reticulum, which are embedded with the ribosome, are known to be rough endoplasmic reticulum. Rough endoplasmic reticulum somewhat is responsible for protein synthesis, and also it helps in the transport of proteins. It also helps in the modification of proteins. So let's see the further functioning of endoplasmic reticulum. See, endoplasmic reticulum is the largest single membrane bound intracellular compartment. It is having an extensive network of closed and flattened membrane bound structure. Enclosed compartment is known as ER lumen. ER membranes are differentiated into rough and smooth endoplasmic reticulum, which you can see in this smooth endoplasmic reticulum and rough endoplasmic reticulum here, depending on the presence of the ribosome associated with their cytoplasmic surface. Regions of ER that lack ribosomes are known as smooth endoplasmic reticulum. This smooth endoplasmic reticulum helps in synthesis of lipids, and also they are called as transitional endoplasmic reticulum. Just make sure that you know about ER lumen here. So the enclosed compartment is known as ER lumen. Lumen means the inner space, inner fluid filled space is known as ER lumen. When cells are disrupted by homogenization, the ER breaks into fragments and reseals into small vesicles called microsomes. So here you can see the endoplasmic reticulum, which are of segments or are of a type of layer. When it is homogenized, it forms a round spherical structures called microsomes, which are small vesicles. Okay, they are known to be microsomes. Microsomes, which are derived from rough endoplasmic reticulum studded with ribosomes, are known to be rough microsomes, and microsomes lacking that ribosomes derived from smooth endoplasmic reticulum is known as smooth microsomes. Easy to remember. Functions of endoplasmic reticulum. First is protein synthesis. As it is embedded with the ribosome, it helps in the protein synthesis. And also here you can see co-translational translocation. What is it? Just remember, co-translational translocation means where translation process and translocation process are happening parallelly. In the lumen of RER, 5 principal modifications of proteins occur before they reach their final destination. These 5 modifications are addition and processing of carbohydrates. Here N-linked glycosylation happens, that is N-acetyl glucosamine. N-acetyl glucosamine, just remember this, N-acetyl glucosamine. You can remember it as NAGA. This N-acetyl glucosamine is added to the amino acid of the protein, will be dealt in the further slides. Then as formation of disulfide bonds. Disulfide bonds happens to be like this. This bond is formed between the chains, and proper folding of proteins happens. If there is no proper folding, then the protein is degraded with the help of lysosome. And next is specific proteolytic cleavage. Wherever the cleavage is needed, wherever the cut is needed, the amino acids are cut in a specific part and assembly into multimeric proteins. These are the modifications, 5 principal modifications happening where? Happening in the ER before they reach their final destination. As I said earlier, smooth endoplasmic reticulum acts as a site of lipid biosynthesis, detoxification, and also calcium regulation. Now let us learn each function in detail. N-linked glycosylation. What does the glycosylation term means? Attachment of a sugar molecule to a nitrogen atom in an amino acid residue of the protein. In RER, this process involves the addition of a large preformed oligosaccharide precursor to a protein. Preformed oligosaccharide precursor to that is referred to as NAGA, that is N-acetylglucosamine. Let us see how. See, the precursor oligosaccharide is linked by a pyrophosphoryl residue to dolicol. Dolicol can be called as a carrier. It acts as a carrier for the oligosaccharide. It is firmly embedded in the rough endoplasmic reticulum membrane. Here you can see the amino acid, right? The N-acetylglucosamine, this N-acetylglucosamine, mannose and glucose, this is a preformed oligosaccharide which I mentioned here. Preformed oligosaccharide is linked to the or attached to the nitrogen atom in a amino acid residue. This is amino acid. It is attached to the asparagine side chain. The structure of N-linked oligosaccharide is galactose 3. You can see galactose 3, mannose 9 and galnate 2. 3 glucose, this is glucose, 3 glucose, 9 mannose and 2 N-acetylglucosamine. It is the structure of this preformed oligosaccharide. This is glucose, okay? Just don't get confused. Biosynthesis of oligosaccharide is not much important, but just remember or just see this. N-acetylglucosamine, 5 mannose residue and also dolicol phosphate is there. They both form first sugar N-acetylglucosamine will link to the dolicol by a pyrophosphate bridge. Then what happens? It activates the oligosaccharides for its transfer from the dolicol to an asparagine side chain of a nascent polypeptide on the luminal side of the RER. So here in the RER lumen side, the polypeptide, nascent polypeptide is activated or the oligosaccharide is activated. Then 7 residue dolicol pyrophosphoryl intermediate flipped to the luminar phase. So the dolicol attaches with the pyrophosphate to pyrophosphate bridge through pyrophosphate bridge. The N-acetylglucosamine plus 5 mannose residue is attached to the dolicol in the ER lumen. Then it flips to the it is flipped to the luminal phase. So here it is in cytosol, right? In next image you can see that this unit will be flipped inside the ER lumen. See here the overall unit is flipped into the membrane ER lumen. Remaining 4 mannose and 3 glucose residue are added. Here mannose 5 is 4 mannose is added and 3 glucose is added at the time in the luminal side after the flipping happening here. Next, entire glucose 3 mannose 9 N-acetylglucosamine oligosaccharide is transferred en bloc from the dolicol carrier to an asparagine residue on a nascent polypeptide, a reaction catalyzed by oligosaccharyl transferase. So this unit is then transferred to the asparagine residue. As I showed in the previous picture, here this before attaching this unit, it will be separate and this will be separate. First the biosynthesis of this oligosaccharide takes place, then in next stage it attaches to the asparagine side chain. Okay, I hope you got to know. See, the oligosaccharyl transferase will help to link that preformed oligosaccharide to the asparagine side chain of the Protein N-glycosylation site it is having N-glycosylation site where the lipid linked oligosaccharide is anchored to that side chain. But the enzyme which is playing the role is oligosaccharyl transferase. Where it is happening? It is happening in the ER lumen. Then function of glycosylation. Why this glycosylation happens in the ER, or what is the main function of this glycosylation is some proteins require N-linked oligosaccharides in order to fold properly in the ER. N-linked oligosaccharides confer stability, protease resistance on many secreted glycoproteins. It is very important for cell-cell adhesion. Oligosaccharides present on the glycoprotein serve as antigens. So antigens have this type of glycosylation. Cell coat or glycocalyx. Have you ever heard about this cell coat or glycocalyx? Especially you have heard in bacteria. So carbohydrates as oligosaccharides occur both as glycoproteins and glycolipids. Plasma membrane also contains integral proteoglycan molecules with surface exposed polysaccharide chain. The term cell coat or glycocalyx is used to describe carbohydrate coating on the cell surface. This cell coat helps to protect the cell surface from mechanical and chemical damage and also mediate specific transient cell-cell adhesion event. So in all these you can see the addition of carbohydrate or oligosaccharide here, which is the function of ER, where it is N-acetyl or N-linked glycosylation. Formation of disulfide bond. It occurs in ER lumen. It is also one of the function of ER, where the formation of disulfide bond happens in the ER lumen, which is facilitated by the enzyme protein disulfide isomerase PDI. It also catalyzes the rearrangement of disulfide bond. Disulfide bonds do not form in the cytosol due to reducing environment, which maintains cysteine residues in their reduced SH state. Next, protein folding. It is assisted by the chaperone. This protein folding is the major function of the ER. Let's see how it happens. It prevents segments of the nascent chain from misfolding. So folding of protein happens to be a major event for the functioning of that particular protein. Important chaperone of HSP heat shock protein is a 70 family located in the ER luminous BIP. BIP means binding protein. BIP binds newly synthesized protein as they are translocated into the ER and maintains them in unfolded state. Two other chaperones like calnexin and calreticulin prevent premature and incorrect folding of segments of a newly made protein. Calnexin located in the ER membrane and calreticulin located in the ER lumen, calcium binding lectins are the calcium binding lectins, carbohydrate binding protein. To understand this protein folding, you need some of the major important points here: calnexin, calreticulin, BIP, everything. To understand it, let us dig into this image. See, here you are having an amino acid, right? And here the precursor oligosaccharide is inserted. The previously I learned thing, no? Galnac, and also there were mannose and glucose. The overall precursor oligosaccharide was inserted, right, to the asparagines thing. Here, after that, glucose is trimmed. Here you are having 3 glucose, right? 1, 2 and 3. 2 glucose are removed. After that, the whole part goes into binding with the calnexin. If it is correctly correctly folded, it gets bind, and it takes off another 1 glucose with the help of glucosidase, where you can see the absence of glucose, right? The N-linked oligosaccharide is present. If this is folded correctly, it will exit from the ER and goes to the Golgi. Next step. If it is not folded correctly, it again goes back to the cycle of glucose. So there will be addition of one monosaccharide here. With the help of glucosyl transferase, there is addition of glucose again, so that it can get again cleanly folded. If it is cleanly folded, then it comes with the comes to the binding of calnexin. Calnexin is, see, it is a chaperone. It prevents premature and incorrect folding. It prevents, if it is incorrectly folded, it gets back to the cycle. If it is correctly folded, then it will let the protein to go further. See, calnexin is located where? It is in the ER membrane. Calreticulin is located where? It is in the ER lumen. Both are calcium binding lectins. See, the overall thing what I explained here, it is in flowchart type. Calnexin and calreticulin are two chaperones which help in preventing the misfolding of the proteins. See, bind selectively to certain end-linked oligosaccharide on growing nascent chains. Recognizes end-linked oligosaccharides that contain a single terminal glucose. Bind protein only after 2 of the 3 glucose that are initially attached have been removed by the ER glucosidase. It's done. Then when third glucose is removed, the protein dissociates from its chaperone and can leave the ER to Golgi. Thus, the binding of calnexin and calreticulin to the oligosaccharides on incompletely folded polypeptide helps in retaining it in the ER. If there is any misfoldings, it remains back in the ER itself. The enzyme glucosyl transferase, what is the function? It adds up the one glucose to the incompletely or completely incompletely or miscompletely folded state of protein. If protein is incompletely folded, the enzyme transfers a new glucose to end-linked oligosaccharide, renewing the protein's affinity for calnexin or calreticulin and retaining it in the ER. The cycle repeats until the protein has folded completely. See here, calnexin cycle translocon is what it is a gateway. The nascent protein chain, it is an unfolded state. There is a disulfide bond added, and there is what glycosylation happened. After that, it is in lumen, right? It is in lumen. Here it is a ER membrane. It is a cytoplasm. Here the glucosidase 1 and glucosidase 2 enzyme will release the 2 glucose. You can see here the absence of glucose, right? There is single glucose here. If it is correctly folded, it goes to the calnexin for the binding. Then there glucosidase 2 will release another one glucose. If it is correctly folded, then this protein will be exited from the ER lumen directly to the Golgi. If it is incorrectly folded, it gets back into the cycle. Or even if there is a fate of this incompletely folded protein towards to where it degrades with the help of some proteasomal enzymes, and it vanishes completely with the help of reverse transport into cytoplasm for proteasomal degradation. Synthesis of membrane lipid. The SER produces most of membrane lipids, including both phospholipids and cholesterol. Phospholipids are amphipathic molecules, and their synthesis from fatty acid CoA and glycerol 3-phosphate takes place at the cytosolic leaflets of ER membrane and are catalyzed by membrane associated enzyme. So this function will be dealt in the next class, as it is of complex steps here also, so it might get confused for you, so it will be dealt in the next class. Thank you.